So we'll start with the first question. So the genomic organization in prokaryotes is called nucleosome, uh, nucleoid, or chromatin, or chromosome. So what will be correct? Okay, so what is Um, what is nucleosome? Can someone tell me what is nucleosome? No one is there. Fine. So basically, nucleosome is known as the structural unit of the DNA. So nucleosome is kind of like packed. So if it is if the DNA is like that, it's goes and a nucleosome form. So when DNA is like packed by clustering of histone proteins, which one like H1, H2, A, H2B, H3, and H4. So these are the structural proteins of the histone proteins. Okay, these are known as histones. Okay, these are known as histones. So histones are the protein that are like used for chromatin compaction. Those are structural proteins or nucleosome proteins. So this histone proteins so this four histone protein H2, H2B, H3, and H4, they first attach to the DNA. Then after that, the H1, which is another histone, this octamer, this is again wrapped out by the H1. Okay, this octamer. So these four, so 4 into 2 is a eight total proteins, which is that's why it's known as octamer. So this histone octamers first wrap around the DNA, and then the H1 histone also then wrap around on that octamer to form the nucleosome assembly. So nucleosome is basically the structural unit uh, of the DNA, okay, or the chromosome. Sorry, the structural unit of the chromosome, rather, I should say. Yeah. Okay. So nucleoid, yes, nucleoid would be the correct answer. So because in the bacteria, if this is a bacteria, you know that there will be a cell wall like this. There will be a cell wall here. Then there'll be the plasma membrane, the cell membrane, and inside that there will be a nucleoid, right? Like this kind of shape, which will be like the primitive, which can say is the nucleoid or the primitive nucleus. Okay, so this will be the primitive nucleus or nucleus. So nucleoid is the correct answer. So chromatin, as you all know, chromatin is the DNA, like the mixture of DNA and protein. So chromatin, what is chromatin? So, oops, sorry. So what will be the chromatin? Chromatin is the chromatin will be the mixture of DNA and protein. So that is chromatin. And chromosome is like the full DNA when it is get compacted. It is known as chromosome. So like humans have total 46 chromosomes. Among that are 22 pairs of uh, autosomes, correct, and one pair of sex chromosomes, correct. which will be for male. For male, it will be XY. For female, it will be XX. Okay. So these are the chromosomes. Delete all those things. Okay. So let's go to the next one. Okay. See what this is. Okay. Which of the following organelles have double membrane complex? Okay. So, okay, please someone tell me that what is the, and what would be the answer? Which one will be the double membrane complex in the membrane? Ear, Golgi, chloroplast, or peroxisomes? Anyway, so let's see. So ear, what is the ear? It's better. I'll just do a screen sharing instead of. Better I just do a screen sharing, but I'm facing a problem here. It will be better. Yeah, this is much better. Okay. Um. So what is ER? So if we see one cell structure, so there in the middle there will be nucleus, nucleosome. Then there will be endoplasmic reticulums here. Okay, right? Reticulums here. Then there will be Golgi bodies also, right? Like this, Golgi bodies will also come. And what else? We'll have mitochondria, of course. So how the mitochondria will look like? This, if I say animal cell, mitochondria will look like this. There will be those crystes here, correct? Membrane fold, crystes. This is the outer membrane, inner membrane. So here also one of the organelles that is double membrane is mitochondria, correct? Mitochondria is double membrane. Okay. Next, what all are there? We'll have cytoskeletons, of course, but I don't draw it here. So we'll have a number of mitochondria, endoplasmic reticulum, Golgi. Anything else I'm missing? Peroxisomes, right? So let's see. This is like the peroxisomes will be like small, very small organelles. There will be peroxisomes, and then there will be lysosomes, correct? So there will be this vacuole like rhizosome, vacuolar lysosomes. And in case of plant, instead of mitochondria, there will be chloroplast like this. So I'll use some other color. Okay. Um, so there will be these types of chloroplast, right? So chloroplast will be also a double membrane organelle. So inside that there will be outer membrane, inner membrane. Inside that thylakoids will be there, like coin-like structures will be there. So this will be chloro. Sorry, that. So it will be have chloroplast, correct? We will have chloroplast. So chloroplast is the correct option here. Now we have now next questions. Okay. Uh, so the bond created by lysozyme in bacterial cell wall between N-acetylglucosamine and N-acetylmuramic acid will be what? Beta 1,3 glycogen, beta 1,6, beta 1,4, and beta alpha 1,4. So this beta position or alpha positions are like it's like those things will be taught in the few classes, and you will get to know that like why what kind of like what how does it look like, okay. uh just so in this case the correct answer is, of course, you know, the beta 1,4. So how beta 1,4 look like, I'll just better show you one. I'll show you one image in there here, then that will be better. So I'll type like beta 1,4 glycosidic bond. Yeah. Yes. How does it look? I'll just show some easier image. Yes. So see, this is how the alpha glycosidic bond looks like, and this is the beta glycosidic bond looks like. So here, both the H here are on the same side, but here the H is on the opposite side. That is the beta glycosidic. If I just show you NAG and NAM, like N-acetylglucosamine, yes, it's wrong. This is beta. It's wrong. Yes. Right? There's the NAG, N-acetylglucosamine, N-acetylmuramic acid, and N-acetylglucosamine. So this is a beta 1,4 glycosidic bond. So between two glucose molecules, the bond is known as the glycosidic bond. In next session, mostly these things will be covered. Okay. Uh, yeah. So okay. So yeah. So correct answer is beta 1,4 glycosidic. That's okay. The K and O antigens, which acts as a surface marker uh, of virulence. What is the what is the like from where it comes? It's from lipopolysaccharide layer, lipoteicholic layer, or it's a inner periplasmic space or outer periplasmic space. Okay, so yeah. So of course you know this is like not this the antigenic layer is the lipopolysaccharide layer, and yeah. So lipopolysaccharide or LPS. It's a very interesting molecule. So I'll tell you a brief like how actually LPS and how actually LPS led to discovery of the toll-like receptors and how it got a Nobel Prize. Okay, so this LPS, so LPS is like it's very commonly it's common antigen molecules. Okay, antigen, it's a common antigen molecule. It's also known as PAMPs. LPS is also known as one of the PAMPs. PAMPs is known as pathogen associated molecular patterns. So these are known as PAMs, okay? Pathogen associated molecular patterns or PAMs. So what it happened? Uh, the discoverer of TLR, I am what is his name? I Hoffman, yeah. No, Hoffman is the dendritic cells. I'm forgetting of. So this TLR got Nobel Prize in 2011 along with uh, Jules Hoffman and Bruce, along with Bruce Butler and Jules Hoffman. Okay. Um. So yeah. Yes. So Bruce Butler was the first person who had got Nobel Prize for discovering the toll-like receptor. Okay, and Jules, along with them, Jules Hoffman was also Jules Hoffman and Rolf Steinman was also a part of the Nobel Prize. Steinman discovered dendritic cells and its role, and uh, Hoffman or Butler, those two, like discovered the toll-like receptor. Okay, so what Hoffman did? So Hoffman was the person. So Jules Hoffman, I was correct only. So Jules Hoffman was the person. So Jules Hoffman, what he did was, um, like he had two types of mutants. So there were like something known as toll. Okay, toll receptor. So they had two types of leaf. It's like toll plus plus, like they are wild type flies or wild type, or toll mutants. Okay, toll mutants. Okay. Now both of these flies, he has infected with some bacteria or fungi. Okay, he observed that wild type flies after the infection was perfectly fine, like they were happy. But toll mutants, they had gone severe bacterial infection and they were not able to survive. So from that, he had thought that okay, this toll, a toll-like receptor, would be the as like it's used as a no, so it is used as another term is, so I'll just wipe it. So it is used as the PRRs. PRRs are PRRs or pattern recognition receptors, correct? So PRRs or pattern. So they are a pattern. So there are patterns. Now this pattern will be recognized by these PRRs. Okay. And later it was found out that was LPS was one such PRR which was detected by TLR4, which is a pattern. And there are like toll like receptor pathway, which is very important pathway in innate immunity, which has got Nobel Prize for that. Okay. So next slide. Next. So this is the lipopolysaccharide. Okay. Next is this. Which of the following species have a linear plasmid? Now this is like very mugging up questions. Like there is nothing much to solve here. Like, uh, this- very like very mugging up question. I'll just okay. So which of the following have a linear plasmid? Paracoccus dentrificans, Bradyzobium elcani, Brucella canis, Streptomyces lividians. The correct answer is Streptomyces lividians, but is like totally marking up. Okay. So during plasmarization, the reagent used for deprotonation is potassium acetate, phenylchloroform, isophenylchrome isomeric, sodium acetate, 0.2 normal NaOH and S2. Now the plasmid isolation method, whatever sir has told was like the alkaline lysis method, right? So alkaline lysis method had like different solutions, right? Solution 1 and solution 2, right? Correct. So okay. So solution 1 contains like glucose, solution EDTA, like tris EDTA, this loses a buffer, and yeah. So glucose, tris and EDTA. So what does its work? So glucose provides the like prevents the osmotic lysis, correct? So it inhibits rather the osmotic lysis. So it maintains the osmotic pressure basically, glucose. EDTA, it's a chelating agent, correct? It's a chelating agent. So chelating agent will chelate any divalent ion. So it will chelate the calcium ions from the cells. Tris acts as a buffer in the cell. Okay. So the next solution is, so this is the solution 1. The solution 2 is NaOH and SDA. So what does NaOH does? Does is make the solution alkaline, correct? And this force like dissolves the membrane. So it's like alkaline lysis. That's why it dissolves the membrane. Now, at what does it does? It's like it makes the pH at around 12. Makes the pH is around 12, and in this pH and this alkaline harsh environment, there are two things happen. That the genomic DNA, it's got degraded. However, the plasmid DNA, it's sheared like. So it is denatured but not degraded. So just denatured but not degraded, correct? Then we can clear the slide. Then we add the solution 3. Solution 3 is potassium acetate. Which is potassium acetate. So potassium acetate, now it's alkali, right? So alkali means there will be lots of OH minus ion, correct? Because I'm giving OH. So potassium will have a K plus ion, right? So this K plus ion will uh, neutralize the uh, neutralize the solution, right? Neutralize the solution, and because of this neutralization, all of this like they will form a acetate compound with, and all the SDS debris and all will get precipitated like a white precipitate. Okay. And okay, and yeah, so then this precipitated, so these things are then precipitated out. Now the next step will be deprotonation. So deep protonation will be done by phenol chloroform and isoamyl alcohol. Isoamyl alcohol. Okay. So this step is done by phenolchloroform isoamyl. So in this test, due to this phenol, all the proteins will be precipitated in the phenolchloroform. All the proteins will be precipitated, and there will be a clean layer. There will be a clean layer. Uh, that will be your plasmid DNA. Okay, and in the after that you add um, alcohol. So since this is a DNA are in a positively genus are positively charged, right? So due to that alcohol, all everything will get precipitated, and that's why we use the alcohol and the last step to precipitate the DNA, and then we redissolve it. The biomolecules synthesized inside the nucleus is histones, DNA polymerase. Is it actually it will be the tRNA that is synthesized. The histone is again a protein. So translation, these all are proteins, right? These all are proteins. This is only the RNA. So there is a general phenomena. Just remember this, that translation or protein synthesis takes place always in the cytosol. Okay, and RNA synthesis, any RNA synthesis takes place in the nucleus. So these RNAs are synthesized in the nucleus, then they are transported out to cytosol, and there is translation. So it is mainly done because of to have like hypothesized to have a proper control over the translation and transcription machinery. Okay. The major proteins that condensation of eukaryotic chromatins are, of course, histones. Because I just now told in the first slide I have explained you DNA polymerase, RNA polymerase. These are like replication and uh, transcription. Ribosomes are used for protein translation, right? The cellular organelle that cannot be obtained intact during the fractionation is endoplasmic rhizome, peroxisome, nucleus. So lysosome is a small, again the when during yeah. So during centrifugation there will be lots of centrifugal force, right? And you think like you have a gel and you are giving like lots of centrifugation force. What will happen if this a gel is a big structure? It will shear it out. So among them, lysosome is a small molecule, small like organelle. Peroxisome is also nucleus. There is a big organelle, but still it has like a proper structure to maintain. So however, in the endoplasmic reticulum, it has only a single membrane structure. In that, it has like very long single membrane structure, like uneven single membrane structure. That will have lots of force and it will get sheared and form small particles of ER. Okay, so ER cannot be obtained with intact during the um, this, during the centrifugation. Correct. Okay. Next. So which phase of the Golgi complex sorts lipid and the proteins? Of course, the maturation phase, because at that phase, all the proteins and lipids are like properly processed and like do the all the post-translational modifications are done. And then in the maturation, from there it is sorted out, correct? So this is the maturation phase. It matures here. The proteins and lipids are sorted and matured. The orientation of proteins in plasma membrane is predetermined by their orientation of ER, nuclear membrane, mitochondrial membrane, or vacuolar membrane. Now, this is a very interesting concept. Okay, so now it is okay. I'll not explain it here because it's quite. complex. Okay, I'll use like why my whiteboard is not working. Not sure why my whiteboard is not working. Okay, so yeah. So what happens? So the concept of signal peptide you have, right? You have so something known as signal peptide, right? Some concept was a signal peptide. Now it's a very interesting and very important aspect in the cell biology. Okay, so what happens here is now, so this is my ER membrane. Okay, I'll use some other color. So say this is my ER membrane. Okay, take a membrane, my ER membrane. This is my endoplasmic reticulum or ER membrane. Now what happens? Now you think and the whatever is the white places you are seeing is the cytosol. Okay. Now there is a mRNA here. Just use some other color. So let's say this. Oh, here I can use colors. Yeah. So let's say this is an mRNA like this. It has, and on that mRNA, the proteins are getting synthesized like this. Ribosomes are there. Correct. Ribosome is there. Now what happens? Uh, the signal peptide. So now proteins, as you know, proteins there are two terminus, correct? One is known as N terminus, one is known as C terminus. Now protein is always synthesized from the N terminus towards the C terminus. Okay, this kind like this, it always gets synthesized. Okay, so now the when the proteins is like nascent protein is coming out or some other color, say when the nascent protein is coming out from here. So there will be a small part here which will be known as the signal peptide. Correct? This is the signal peptide. Okay. This is a small part. What this will do if it has an ER signal peptide, this thing will go and bind to something known as SRP or signal recognition peptide. What it will do is this will go and bind here. What it will do is it will form something like this. Okay, so again, if I just draw the membrane here, okay, if I just draw the membrane here, and if this is the mRNA, and this is my ribosomes, this thing will take the whole thing here. Okay, that's why it is called like. So I'll come to here. Okay, that's when the then what happens? This signal peptide, there is a signal recognition SRP is here, right? When the nascent protein is coming out, the signal peptide will come here and it creates something called like a pore. Okay, inside the membrane, it creates some kind of a pore from where the nascent protein, which is like this, it's start going inside the ear membrane, ear lumen. So these are my cytosol and these are my ear lumen. Okay, ear lumen. See, it comes inside. Now there are other process, and if there is some portion here which have a hydrophobic region, then what we'll do after next step? After next steps is after next step what happens here is in that membrane protein. So now this is the protein here. This is my protein. Okay, and inside that it has this hydrophobic loop which has been like transferred here. Now, as I always told you, protein is always synthesized from the N terminus to C terminus. Now think if this protein is forming a vacuole, see this part of this is forming a vacuole like this. It will be it will be a single membrane, of course. That's why I'm just not just wanted to make it bigger, so that's why it's a rubber membrane. So this now when it's coming out from the ear as a vacuole and going to that fuse there. Okay, so this orientation will be maintained because then this part will get to fuse here and this part would to get here. So whatever orientation is here, it will get maintained here also, right? And if the protein was somehow, there are other mechanisms where the proteins can also become as like the opposite directions also, like it will not synthesize opposite direction, but it will be put inside as a opposite direction. Like if there is C and this will be N, then also it will go and bind there. So then it will be C and D. So the question was that orientation of the proteins in plasmids are predetermined by their orientation of course, the endoplasmic reticulum. If it is oriented C to N in the endoplasmic reticulum, its orientation will be C to N. If it is N to C, then it will be N to C. Right? So like that. Okay. Okay. So the next question. The membrane carbohydrate are exclusively confined to the cytoplasmic phase, core of exoplasmic phase, core of the bilayer, or the both side of the exoplasmic phase. So, so do you remember that there was an image by sir what so shown that there was like say not very good at drawing, but say this is my like bilayers. We have these things here, correct? Like that, we have this phospholipid here, and all the lipid chains were here, right? If you remember, there were some integral membrane proteins, some outer membrane proteins, correct? So there were some protein that had this carbohydrate. So you tell me that which one is that? Uh, which one is that particular phase? Okay, which one is that particular phase? Uh, which will have the So did you able to remember which one was this thing? So yeah, it was on the exoskeleton phase. Now one you can think of this core of bilayer will never happen because carbohydrates are polar molecules, right? So carbohydrates are not like hydrophobic molecules, right? Carbohydrates are always hydrophilic molecules, correct? So they cannot be in the core of the biplayer. If both sides of the exoplasmic or this phase, they cannot be both sides of the exoplasmic or phase because of course the uh, if both sides of exoplasmic or endoplasmic phase happens, it has to pass through the bilayer. Now since it's a hydrophilic, it can't pass through like that. So cytoplasmic phase or exoplasmic phase. Now you know that this carbohydrate tagging or glycosylation, what is known as, it's formed in the Golgi. When Golgi it is formed, it itself is like a process, and after the processing, the glycolized. Okay, I'm sorry. What the instruction? Okay, yeah. Yeah. So it cannot be now it's formed and the glycosylated like the peptide is actually on the outer surface. So when it goes and fuse with the vesicle, it will actually be in the exoplasmic state. Okay. So this again you have to remember this thing to be. So yeah. So the marker for lysosome is which one? Uric acid, cytochrome oxidase, sialic acid transferase, or uric acid is it's not it's a metabolite. Cytochrome oxidase, as you know, it's present in mitochondria because it's a part of that ETCs, electron transport chains. Sialic acid transferase again is just like uh, it's present in mainly in the Golgi. It's a glycosylating enzyme. Cathepsin, it is one of this lysosomal cystine protease. So cathepsin is the correct answer. Okay. Transferrin, soluble carrier protein that transfers ion through the blood by a process known as phagocytosis, pinocytosis, cryptomediated endocytosis, or exocytosis. So is a transferrin receptor. This always has a transferrin receptor. Again, it's something that you have to remember. There is nothing to be like. Transferrin has its transferrin receptor. So whenever there is a receptor, there will be a receptor mediated endocytosis. And one more thing you can remember is that uh, our uh, this plasma membrane is a amphipathic, right? So it has a hydrophobic layer. So it's like a hydrophilic chain, and along with that, it's like a hydrophobic lipid molecules, right? So any charged particle like Fe2 plus ions cannot pass through, sorry, any hydrophilic molecules like ions cannot pass through this layer, correct? You always need a transporter, and transporter is there if either there will be some receptor mediated endocytosis with some transport, it will the ions will be bind to some transporter, or it will have a ion channels like sodium potassium ion channels. Okay, again the lumen of the pH of the late endosome. What is lumen? Again, it's a very mugging up question actually. So one thing I can just tell you to just remember this particular thing. So if this is, say, this is my cell, okay, so this is my cell, and so this is my nucleus here, and I have some lysosome here, okay, this is my lysosomes, okay. Now whenever there is a receptor mediated endocytosis happens now, so whenever there is a receptor mediated endocytosis, this is my vesicle formed, okay, and it has that microtubules, so tubule layer, so it will go to here. Now it always had this pumps known as H plus pumps. So when though this kind of endosomes are known as early endosomes, and as it goes on matures, it forms late endosomes. Okay, and then it can go to fuse with either lysosome or other part of the body, but most of the time it will go to lysosome. So this H plus pumps, what it will do, it will always pump in, it will always pump in H plus ion inside for the derivative. So always, if the pH here is say 7 to 7.4, always it will go on, because H plus ion is pumping up, it will go and become more and more acidic. So 7 to 7.6 of course don't have, because late means it is near the lysosome. So it will be 6 to 6 to, it will not be like 6 to 6.2, it's like medium endosome. 2 to 3 will be too acidic, which is like go to the lysosome. So that's why it's like around 4.5 to 5.6, which will be the correct answer. This somehow you can guess the answer, but yeah, anything you have to remember. The decision to divide is taken in the, of course, you always all know it's a very okay, G1 phase. So as sir told, if we have a cell cycle, we have several phases of cell cycle, right? So the biggest phase or is the preparation phase is the G1. Then there will be the synthesis phase or SGS. Then there will be G2. Actually, my drawing is not correct. It's like 4, but mitosis will be like the smallest portion will be the mitosis, and G2 will be the biggest portion. So if I just do it correctly, I just do it correctly. Maybe this will be the G2 and this part will be the correct. So the cell cycle goes like always this orientation. It never goes back. There will be a checkpoint. G1 checkpoint, G1S checkpoint, G2M checkpoint, correct? So yeah. So uh, G1 during the G1 phase, the cell decides that okay, should I divide or not? Should I have enough resources to divide? So do I have enough proteins to do? Do I have enough cyclin molecules to do? Do I have enough the organelles to go for dividing? That's why the decision to divide is taken during the phase of cell cycle. And always, there will be another like loop here, which will be the G0 phase. Correct? Histone are synthesized in. Now, histone is a kind of protein. It's a very interesting protein. So histones are mainly synthesized in the S phase, because when this S phase, what happens that there will be a synthesis of DNA, correct? So your like this chromosome arms will be double, right? So like chromosome will look like this, right? Metaphase chromosome, how it looks like, correct? There will be your nucleus, and from there it will get divided, right, in the anaphase. So when you are synthesizing new DNA, that means you need to get it compacted, right? So otherwise, remember we have some gigabytes of DNA, and that comes in so small space. You can think like a gigabytes of DNA is like kilometers of road, right? If you just stretch it out, it will be a long. It cannot be fit inside the nucleus. So what cell does? It compacts. So actually, inside chromatins there are like very highly compacted DNA here. Very highly compacted DNA here. Correct. Correct. In part of the in part of the centromere, right? Centromere. So centromere. This is telomere. Correct. So this is very congested DNA, and more you go towards the center, it is more and more congested. So when you are synthesizing new DNA, I need that to be compacted, right, during that S phase. So that means I need histone proteins, because as I told you, we are nucleosome core proteins. So they need to be compacted, more and more compacted, because it goes to the M phase. Because otherwise, if it just become like this, then it will be very difficult to take that two DNA, separate two DNA out and during the anaphase, right? That's why histones are mainly synthesized in the S phase. That is again needed for to compact the DNA during the cell cycle, correct? The cells which do not divide, once they are differentiated and enter into G phase, lymphocytes. Lymphocytes, you know, all know. Whenever there is infection, there is lots of like white blood cells are they come out. When there's COVID infections, people are saying that this there are lots of inflammations. White blood cells are like going mad. Well, so lymphocytes are there. Epithelial cells of intestine. Epithelial cells of intestine. They are nearly every day on the epithelial cells of your intestine. Even your skin cells. They're every day they're replenished. So if they wanted to replenish, then sorry. If they want to replenish, then they have to divide. So they cannot be in G0 phase. Smooth muscle cells. Smooth muscle cells. Again, it's like a every time you are constructing, so there will be lots of ATP generation, ROS generation, cells will die here, right? So they have to be divided. So the cells that cannot divide is actually the lens cells. More specifically, so my part of my PhD work is in cornea and lens. So more specifically, if I just clears the this, if you more specifically, if you go, if this is our lens, this is our lens. It is not that our lens cells do not divide. There are specific lens cells that do not divide. So there is a cuboidal epithelium here. I hope the epithelial part will be covered in this course. Yes, I can also cover in one of this class. Correct. So this is a cuboidal epithelium course in the in the lens. Correct. Single layer here, like nucleus. What happens? There are cells that gets divided and go like this or like this. And as they go on there, in the middle, they are very constricted cells, and they are mainly form of this one protein that is known as crystallines. Okay, crystals. So lens, if you see a glass lens, this looks like a crystal. If you see a lens, original lens, like I have isolated mouse lens, it exactly looks like a crystal. Let me see if I can show you one of that thing. Okay, I should have one. I should have a picture. Um, Let me see if I can show you one of the lenses that will be quite interesting. Com, yeah. No, it should be here in the. Hmm, okay, I will show you one second. Let me see. I can show. I have the image. Okay. Um, Let me show you here. Ah, this here. Uh, yes, so this is how the eyeball looks like. Okay, and this is how the eyeballs look like. Okay, so this is the lens. So lens, if this I am showing you in a in a in in a face contrast micro, that this part is black because light is coming. So this is how the crystalline looks like. If you see in the eye is like, I can, if I enlarge it, you can see there is like blue thing, and inside there is the lens. Okay, so I isolate this from mouse. This is one of my PhD work basically. Okay, so this the correct answer is lens cells. So during which phase of the cell cycle chromosome become highly condensed to prevent DNA damage and facilitate segregation to opposite poles? So mitosis is or divided in four of the phases, right? Prophase, metaphase, anaphase, telophase, and another last portion, like it's not part of the, to the mitosis, but cytokinesis. Correct. So in prophase, what happens? Cells have different organelles. So they lose this nuclear membrane, all these organelles they lose. Okay. At metaphase, if you see a metaphase cell, like uh, which is under like very condensed chromatin. So like this. This I can also show you a beautiful image. Okay. So I have this. I can also show you a good image of this. Uh, where can I show you? Maybe this one. Yes. No, not this one. Okay. Any work presentation. Maybe here I show you. Let me see. So I want to show you how the actually the chromosomes looks like if you just block them. Okay. See if I have some. Yes. Uh, aha. Yeah. See one of these images. Okay. So see one of these images. Correct. So I think I'll have one more very good presentation. Let's see if I can show you. So I'll have either in HEK or HeLa cells. HeLa cells, you will see the, yeah. So this is the image I wanted to show. See, so this is the cells that are just blocked in between prophase and metaphase. Okay. And it was just going to enter metaphase, but not. So these were treated with the nocodazole, which is a cell cycle blocker. So as you can see, this is the this is one second. So this, see, these are all small chromosomes, and these are like just very constrictively packed, right? Because I'm holding that, I'm saying okay, you stop there, and see they are very constrictively packed. If you see any of the image, these are HeLa cells. Okay, these are HeLa cells, but see here again, very compactly packed DNA. Correct. So like that, this get. Anyway, okay. So this is how it will. I don't know what is happening. Anyway, yeah. So at metaphase, DNA get very tightly constricted. Now in anaphase, of which get very tightly what is called constricted. Now become highly constricted, prevent DNA damage, and to facilitate segregation to the opposite poles. So of course it will be metaphase. Now the answer that is given is anaphase. Okay, but I am not sure it will if it will be anaphase, because you see the chromatin, it will be like this, correct? So anaphase where it segregates, but it's not highly, it's most highly condensed in metaphase. So I think the answer whatever is given is not correct. Okay, pH in the mammalian cell culture is monitored by Texas red, phenol crystal violet, tip type, and blue. It's a phenol again. It's nothing to be like, cannot explain anything. It's just something to be very much like to be remembered. Okay. Uh, so yeah. So these were the questions from week 3, and let's discuss if you have anything else. Okay. Otherwise, I can do something. I can teach you some like some cell biology techniques that we generally use in the lab setup, right? So, so there are lots of things that we do in the lab, right? So, say first, okay. So, what kind of so can you guys can unmute and say what kind of experiments at least you have done in your lab regarding to cell biology, like with bacterial cells or like human cells, whatever cells. Can anybody tell me? Have you done like isolation of the squamous epithelium from your lip and stained it and just tried to see if it you can see them under the microscope, normal microscope? Yeah, I just want to interact with you and get to know what you kind of people do like during our BSc or like masters. I have not done much experience in this kind of like, yeah. If you have done this kind of any experiments or so anything, just two, three things I wanted to discuss with you before I end the session. Maybe I will take another 15-20 minutes. So experiment wise, there are two things I that are very commonly used in different cell biology techniques. Okay. So one thing is wet and wrap. Okay, so before I go there, so I want a hand for this. Do you guys know what is fluorescence? Do you guys know what is fluorescence, like GFP, RFP, YFP? I just want to know if you guys know what is GFP, RFP is like that. Ali Javed, Mohammad Rofi, Rida, Shana, Zainab, anyone? Do you know what is like fluorescence or fluorescent protein? Hello, can anyone let me know? Any okay. So fluorescence I'll just give you like very layman. I'll give you some very layman kind of like uh, explanation. What is like uh, okay. So in very layman language, fluorescence is like if you excite a molecule with light, it gives you back some light, some light of a higher wavelength. Okay. Why this what is happens? No. Now there are some molecules here. So they have this electrons here in different cells. Okay. So there are this electrons here. Here some electrons, different cells, and this is the this is the my nucleus. Okay, this is the nucleus for this. So these are some electrons are going. Okay. So when light is nothing but photons, correct? Photons. Now photons, what is Gavri? It has some energy. So when you give energy to a photon, what happens that if I just draw one more shell here, some electron from this outermost shell jump to this shell. Okay. After that, it again returns back to its previous shell. And that time it again, so now it gained energy and it had to return. Now it's in an unstable state. So now we have to again return back. So this is step one where I'm giving, and this is step two. Now from going from step one to step two, it emits again some photons. Now there are several laws and which shows that this have less energy compared to this one. So as you know, E is equal to H nu, sorry, E is equal to H nu, which is H by lambda. So that's why whatever, so if it has to be less energy, it has to be more wavelength. So if I just draw a graph like this, you may have already also seen in different like feel that okay. So if I excite here like this, my emission will be like this. So see, there is the highest wave. If this is okay, so this is my wavelength or wavelength or lambda, and this is my intensity. Okay, so as you can see clearly, there is a shift from the wavelength, and this shift is also known as Stokes shift. Now, what is the interesting thing about why I am talking about is like this things fluorescence. It is a very interesting thing. No, you can use fluorescence to stack cells, right? To stack sequencing. Now, say I have a say I have a protein A. Okay, I have a protein A. I have a protein A, and I don't know where it goes in the cell. Okay, I have no idea where it goes in the cell. Okay, so I have no idea like if it is go to the if it does go to the nucleus, or if it does go to the say uh, ER, it goes to the lysosome. I don't know, or it stays in the membrane. So I want to know the localization of protein A. I want to know the localization of the protein. Now, how do I do that? So what I can do is I can have a DNA construct like that. Can have a DNA construct with the DNA sequence of protein A, and I can tag this same protein with a like here. Yeah, I can then tag this particular protein or some fluorescence protein, which is very famous protein is GFP. GFP can tag this protein with a very famous protein GFP. Now I put this constant inside the cell, this is my cell, and I after say I put this constant, I made something, and I put it inside the cells, and say after putting inside, maybe after say 12 hours, I start I uh, excite the molecule. Now, since this protein is now, when the protein will form, what will form? It will be like this A protein, and it will be tagged with this GFP, right? Now, if it is tagged with this GFP, wherever this protein is, if it is in the nucleus, I can find it like this, correct? Or I can find it here in the cytosol somewhere. Now, how do you know that now? Now I can. There are other fluorescent dye also. So I have a fluorescent dye X, which is like, which gives me, say, I have a fluorescent dye X, which gives me red fluorescence. Okay, which give me red fluorescence. Now I see, okay, this red fluorescence is coinciding with my green fluorescence. So that means, and I know this goes to nucleus. Okay, this guy goes to nucleus. So now, okay, I'm seeing that, okay, this red guy and this green guy is fluorescing together. Correct? I can see that by different methods of microscopy. I can see, okay, these guys are fluorescing together on the same place. Actually, it will be in the here. They're fluorescing together at the same place. That means that, okay, my DNA, my construct, my protein goes to nucleus because it's fluorescing together in the nucleus. That's how it's very, tagging a protein is very useful to just observe the local. Okay, why does my protein is going, right? Or what my protein is going. Now, from here only, I'll come to the second point. I have told you some two terms, right? At the beginning, FRIT and FRAP. FRIT, known as fluo, fluorescence resonance energy transfer. Now I have told you that during fluorescence, what it happens? I have told you it has to give some energy, right? Now think of like a molecule, two molecules. Now they are very close to each other. Okay, so one molecule is here. Say this gives me the red fluorescence, and another molecule is here, which gives me the green fluorescence, right? What I do was they are now say this protein is I'll just this is tagged with the protein A, and this is tagged with the protein B. Now I want to, my question here is whether A and B proteins interact with each other. Now how do you do that? So you tag two proteins. Now you use a concept of fluorescence resonance energy transfer. What now there could be two possibilities, right? As one possibility is possibility one, that these proteins are interacting. Possibility two is that okay, these proteins though they don't interact with each other. So like there is my B protein, sorry, which is tagged with GFP wala, and I have the A protein, which is like here, which is tagged with the red fluorescence protein. Okay, maybe. Now, now think. Now the concept is such that if they interact, if I give an energy to excite only GFP, then it will emit, and it will emit in say green range. Okay, but I am but if they're very close to each other, it's like few nanometer range, then if I am exciting this protein, this emission itself can excite this red protein. So instead of instead of getting green, I am getting red light. So that means that these A and B proteins are interacting with each other. If I am getting only red green proteins, only green, that means A or B and far from each other. But I'm saying okay, I'm giving excitement to the green protein, but I'm getting emission at the red range. That means that these proteins are interacting. Now, why this is very important? This range, or like say this range, to get like this resonance. So this is resonance, right? This protein I'm giving and this protein I'm getting. So their emission is resonating with each other. So this can be a few. Oops, this can be with the just few nanometers. So think of like 1 millimeter, 10 to the power minus 9 meter, or 10 to the power minus 6 millimeter, correct? Yes, at that range, it's known that they have to be interact. So that's why freight becomes very important tool when you say, want to say that okay, A and B proteins are interacting or not, correct? Now second thing is another very interesting thing. Okay, so how do I explain this? So FRAP, okay, FRAP, FRAP. FLAP stands for fluorescence recovery, okay, after photo bleaching. What does bleach do? Bleach makes all the things white, correct? In this case, photo bleaching means that whatever fluorescent molecules were there, I am giving some very shiny light, and all of these fluorescent proteins are getting inactivated. Okay. Now think of a scenario. Okay. Let's say So let's say you want to calculate the generation. So yeah, so let's you want to calculate the generation time or like production time of a protein. So how will do that? So we want to know that okay, if my A protein when it expresses in like I'm seeing a protein here, but how long it takes this protein to express, correct, or get like in a huge quantity. That could be months, that could be sorry, that could be minutes, that could be few seconds, correct, or that could be hours. Some this is very like control step that what is the translation rate of a protein will do. So how do you do that? Now again you think of a cell, okay, and inside the cell you have protein A tagged with again say GFP, green fluorescence protein. So now you will have this same proteins curve puncta here. So if I just make a nucleus here, so you have this protein curve puncta here some everywhere, okay. Now what you do is you do a photo bleaching step, okay. So you do a photo bleaching, correct. If you do a photo bleaching, what will happen now? There will be no fluorescent protein. Sorry. There will be no fluorescent protein. The cells will just look like this. Now, say you see after 30 seconds, you again see, again you start to see this kinds of puncta. That means in just 30 seconds, my protein is getting regenerated. Okay. Or say, okay, I want to see that okay, at which phase of the cell cycle my protein gets for example, there was one question, right? I want to see okay, histones at which phase of the cell cycle histone proteins get uh, histone proteins get synthesized. So how do I know that histone proteins get synthesized? What I will do is what I can do is I can follow the cells through a cell cycle, correct? Now I photo bleach the cells after one M phase. After M phase, I photo bleach the cells. Now there is no M phase. After the M phase, there is no histone proteins which are tagged with GFP. I photo bleach the cells. Now I follow the cells, I follow the cells to G1, and maybe I use some blocker which is like G1S blocker, and after that I treat the cells, and I have another set which I don't use the G1S blocker. So the cells which have that G1S blocker will not go to the S phase, so S phase will not go, but the untreated cells will go to the S phase, correct? And what you see in that phase, now here I have photo bleached. At M phase I have photo bleached the cells. There is no histones with GFP tag. Now I see in the S phase, after S phase, suddenly after G1 S phase, I see in this phase suddenly there is again green fluorescence are coming. That's how I get to know that okay, this protein is only expressed in S phase, not in the other phase. This is how people actually have invented that which proteins, which cyclins have expressed in the which phase of the people have demonstrated like which cyclins. So we all know, right? There are like a few cyclins, right? So there is G1 cyclin, then G1 S cyclin, and S cyclin, and uh, M cyclin or G2 enzyme. So I just remember it like this: cyclin D, Right, DEAB, sorry. I just remember it like this, DEAB, DEAB. So cyclin D for this G1 phase, cyclin E for this G2S, and cyclin A or whatever written, cyclin B, okay. And this side, the CD case, right? Cyclin dependent kinase, 4 by 2, 331, something like that. I forgot that. Cyclin 4. I think 4 by 2, 331, or something like that. Okay. That's how the cyclins are developed. Okay. Yeah. So this is very two interesting things, right? And in cell biology, people almost always do fluorescence and this FRET or FRAPS. Even this fluorescent tagging, the first thing that we do is a fluorescent tagging, correct? So people very much do fluorescence tagging. I'll show you one. I can show you one beautiful picture again if I get that. Of course, I hope I'll have. So I show you something like ah, see, this is a fluorescent thing. So here, what I have done was I have tagged the tags one micro one intermediate filament protein is a cytoskeletal protein with green, and this is my nucleus with stained with DAPI stains with DAPI go and binds with the nucleus. Sorry, the DNA. So see, this is the fluorescence, how it looks and under the microscope, and when you take the images, this is how it looks like. This fluorescence cells, it looks like. There are other examples I am not able to find now, but I will show you that where I have tagged the protein with GFP and see that okay, this protein is here. There are lots of examples. Okay, so yeah, so that's what, like, you know the cell biology, you understand the theory, but then you apply those theory, correct, to get to know more about the cells, do more research, right? And yeah, so I think we can stop here now. So, okay, any questions? Again, I'm asking, any question is there, please let me know. Don't hesitate. Anytime you can stop me and let me know if you have any questions regarding any topic, or you want to explain me any topic, just mail me. I have given my mail ID again. I'm just showing up. This is my mail ID. Just drop me a mail. Okay, sir, I want you to, or uh, Shobik, I want you to discuss. If you can please discuss this particular topic, and if I know that, I will try to discuss. I can tell you one thing, if you ask me to discuss structural biology, I cannot discuss because I have no knowledge of structural biology. But other things related to cell biology and molecular biology, if you want me to discuss, I can try to discuss. If I know what, I can read and discuss on. Okay. So yeah. So that's it for today and for this session. And that's it. So I think we can now stop the sessions, and thank you very much for joining. So I would like I don't know, ask your friends to join because I know there are two sessions overlap. That if they can join, also join. I'll be very happy. There is only 4 or 5 people have joining. There are more people can join. I'll be very happy. So is there any questions? Okay. So if there is no question, thank you very much for joining. I'll end the studio session here. Thank you very much.